Hello and welcome to the Poultry Health Research Network here at the University of Guelph. I'm Ryan Snyder, the coordinator, and I'll be your host today. Today you're going to see an interview from one of our graduate students who's working on poultry research right here at the university. Enjoy. Today we're sitting here with Jake Astill. Jake, thanks for joining us.
You're welcome.
Tell us about yourself, Jake. Tell us about your background and how you came to be involved with poultry health research.
Sure. So I did my undergrad at the University of Guelph, and I was really interested in immunology, in biology, but more so in like human biomedical science. So I came to pathobiology looking to do a master's in something to do with just virology research, and then I discovered that there's a lot of immunology and virology research to be done in agricultural settings. So Dr. Sharif's lab works with poultry, and I found an area where I could work with immunology, do vaccine research in chickens specifically. And that's how I found my way to poultry research specifically.
Fascinating. And so that's where you're working now. Tell us about your role.
So I did my master's, my MSc in Dr. Sharif's lab I finished in 2018 in May, and now I'm a research assistant just working on all the projects in the lab.
Okay, and this is Dr. Sharif's lab in the Department of Pathobiology.
Yeah.
Which group of poultry are you working with?
So all of my research was with layers,
Okay.
But there are lots of other projects working with broilers.
Okay, and what age groups were you normally working with?
For the birds?
Yes.
Yeah, so all of my research was done in layers from hatch to 6 weeks old.
Okay, and what specifically were you looking at?
So I had 3 experiments and different vaccine trials. So basically looking at enhancing the immune response elicited in chickens just by changing the type of vaccine. So we looked at different inactivation strategies and then also used different types of adjuvants.
Okay, and what were some of the major findings?
So one main thing we did was we changed the way we inactivate the virus. So we used different types of chemicals to kill the virus to put in the vaccine, and we also used types of radiation, so gamma radiation. And we showed basically that by using different types of inactivation strategies, this changes the immune response that's elicited in chickens.
Okay, and so let's take a step back for a second. Mm-hmm. The viruses and the pathogens that you've been working with are issues to the poultry industry in what fashion?
So in some parts of the world, poultry are infected commonly with influenza viruses. So in these areas of the world, this can affect production, potentially in layers. We work specifically with an H9N2 influenza virus. It also, poultry workers do come in contact with the virus. So that's just some area of concern as well for poultry workers.
Was there any major collaborations going on with this project between other labs at the university or other institutions?
With my project, not so much, but there are other projects in the lab that.
Okay, can you tell us about those?
I don't know the details specifically of the collaborations. Maybe you'll email or interview Khaled, maybe or someone else, I'm not sure.
And so what's been the most exciting thing about your project for you?
I just appreciated the fact that I really was interested in immunology and vaccines. Mm-hmm. And the fact that I could, produce a vaccine on my own, make a formulation, and then working with poultry, you can handle the birds yourselves and administer vaccines and perform this research and, see the outcome of your vaccine. Mm-hmm. So I think it was just really fulfilling to kind of see the whole thing, make a vaccine, see the response.
Yep, and so take us kind of through one of these trials. So what would be involved in terms of the animals and the facilities that you used and the materials that were done?
Sure. So it starts with producing the virus. So to produce enough virus to make a vaccine, we actually grow the virus in chicken eggs, embryonated chicken eggs. It's a good way to grow high titers of virus. So we grow large amounts of virus, and then we would inactivate it in some way. So typically you treat it with a low amount of a chemical. So for example, formaldehyde at 0.02%. Another one is beta-propiolactone. And then we also inactivate the virus with gamma radiation. So after you've inactivated the virus, we just want to determine the dose. So we would measure protein concentration of the inactivated virus and then give the same amount to every bird. So we typically inject it into the thigh, so it's intramuscular. At this stage, we'd also combine it with different adjuvants. After we administer the vaccine, we would basically look for immune response. So we'd look in the serum of chickens, so measure antibody response every couple weeks, or every week for about a period of 4 weeks. And then we also would look at cell-mediated immune responses by taking specific lymphoid organs and then looking at gene expression and protein production in those cells.
Okay. Some of these samples, were you able to process them yourself, or did you have to send them away to another lab?
All the sample processing was done by me or by others in the lab. All the chicken experiments are done in the isolation facility at the University of Guelph. The vaccine production, the whole thing is done, except for the gamma radiation of the influenza virus was performed at the University of Toronto.
Okay. And were most of these processes already well defined in your lab, or are you doing some more exploratory procedures in your project?
So, that's a good question. Typically for vaccine production, formaldehyde or formalin was used to inactivate. So trying BPL and gamma radiation was new. I mean, people have done that with influenza virus, but testing it in the immune response in chickens for H9N2 was something different. That's the main difference there, yeah.
Okay. And so if we to look at your results specifically, how can this, down the line be applied to the poultry industry?
So, I mean, in North America, I don't think chickens are vaccinated against influenza viruses. There are vaccines administered in other parts of the world where the virus is endemic. And typically they would use an emulsion-based adjuvant, like an oil emulsion adjuvant. And we did include that as like a control in all the research experiments. And in most cases, some of the adjuvants we included, so toll-like receptor ligand adjuvants, actually enhanced the immune response to a level that was significantly higher than what would be used with an emulsion adjuvant.
Wow.
So it just provides some knowledge to know that there are ways to formulate the vaccines to enhance immune responses in chickens.
Very cool. And so what's next for you after this chapter of your life? Are you looking to stay in academics, or are you looking to join the industry?
Looking for industry, not necessarily poultry, but that opens up another area of research. So
yeah.
And right now I'm just still in the lab working with all kinds of different projects.
Great.
So yeah, just enjoying that right now.
Yeah. Is there anything else that you'd like to say to the poultry industry or the academic community?
I guess thanks to funding agencies that let me do the research. Thanks for Dr. Sharif for going to get the funding and having me as a student. Did not think I'd be injecting chickens with vaccines ever when I was in my undergrad, but then you just find yourself doing it. It's a great experience.
Excellent. All right. Thank you very much, Jake.
Thank you.
This has been another Poultry Health Research Network student interview.